Hello everyone, welcome back to Doctor Chinny Lectures. I hope you all have been doing well since our last session. I'm excited to have you today because we are diving into an essential topic in immunology. We're going to be discussing antibodies in detail. We'll cover the definition, all you need to know about the antibodies, the structure, the function, the relationship between antigen and antibodies, and so on. Whether you are here to refresh your knowledge or you are here to complete this new subject, I'm thrilled to guide you through it. So grab your notes and let's get started. On our previous topic, I put out a note on antigen where I discussed antigen in the introductory manner. I stated the definition of antigen. I talked about the types of antigen, the location of antigen in the body, and the application of antigen, medical application of antigens. So today we're going to be talking about the antibody. So grab your notes while we start. We are talking about antibody. Now, if I may ask, who can tell me what is antibody? What is antibody? I know a lot of definition will be running through your mind, but I'll give you a key note on how to go about the definition. Before I start discussing antibody, I will tell you different concepts you need to know in the definition. Like, let's state the first thing you need to know about about antibody is that number one, you need to know that an antibody is a Y-shaped glycoprotein. So when I mean glycoprotein, I mean a molecule that is linked to a carbohydrate. That's sugar. So when I say the first concept you need to know in the definition is that antibody is a Y-shaped glycoprotein. Okay, this Y-shaped glycoprotein, you can as well say it is four polypeptide chain. So it's a Y-shaped glycoprotein, that's polypeptide. It is a tetrapeptide chain. So you just say antibody is tetrapeptide chain. It is four polypeptide chain. So you just say it is a four polypeptide chain. Another concept, key notes you need to note in the definition of antibodies is that, okay, if I may ask now, where is the antibody secreted in the body? Which part of the body is the antibody being secreted? I'll help you with the answer. The antibody is secreted at the plasma cells. The secretion of the antibody is at the plasma cells. So now, another question which I'll throw to you now is, which component of plasma cells is the antibody being secreted? It is at the plasma protein. Antibody is secreted at the plasma cells. Which location at the plasma protein? Then, which part of the plasma protein? You just say antibodies are in the plasma, and the part of the plasma protein where it is secreted is at the gamma globulin. So now say is secreted at the plasma cells. I would like to explain these plasma cells in a more detailed way for a retentive memory. Now, I need to draw it. So this is a cell. This cell have a negative charge here and a positive charge here. And we have the plasma cells located here towards the negative side. So if I ask you where you can find the plasma cells, just note that the plasma cells is located towards the negative side. You see that the plasma cells tends to move towards this positive side. The plasma cell located towards this negative side tends to move towards this positive side. The movement is known as electrophoresis. So the movement of this plasma from this negative to the positive side is electrophoresis. Now, the question now is, during this movement, are they moving slowly or are they moving faster? So the answer is that this plasma protein, they are so heavy. They are so heavy that they tend to move slowly. The heavy ones will tend to be towards this back, at the back. So if I now ask you, what weight can you say this one at this end, this one towards this positive side, has small molecular weight, and the one that tends to be behind this smaller ones, they are the heavy ones, they are the heavy chains, and they have large molecular weights. So this small molecular weights, you can call this one albumin. This is the albumin. And these larger ones that couldn't move so fast, that was moving so slow, you call these ones the globulin. So this globulin now, in which part of this globulin can you find the plasma cells? This globulin has the alpha, the beta, and the gamma. So what part of this is it, at the alpha or beta or gamma? Where do you find the plasma proteins? So you see the plasma protein at the gamma region. So you say this plasma proteins, I need to highlight it so you can remember. So this plasma protein, you'll find them at the gamma globulin. So you say that this plasma cell, when they tend to move, they are so heavy and they move slowly. And the smaller ones tend to move faster. They have the small molecular weight. The heavy ones, they are the globulin, and they have this globulin, we have the alpha, beta, and gamma. And the plasma protein are located at the gamma region. That is why we say antibiotics are gamma globulins. Okay, let's move forward. We can say the plasma cells are specialized white blood cells that play a crucial role in the immune system, that secrete large amounts of antibiotics. So these plasma cells, they are specialized white blood cells. So plasma cells that leads to these antibodies are the B cells. They are the B differentiating cells. So the B lymphocytes, they encounter an antigen that matches its specific receptor. It then activates and differentiates into a plasma cells. So when these B cells, the B cells in this plasma cells, when the B cells encounters any antigen, they tend to activate the system. They tend to activate the system and then differentiate into plasma cells, thereby releasing their contents into the blood cell, releasing antibodies into the bloodstream to fight the antigen. So we already noticed that we stated that the antibodies, they are Y-shaped glycoprotein, they are polypeptide chains that are secreted by the plasma cells. Now, another thing I want to tell you today is that there are different names you can give. There are different names given to this antibody. Antibody, it can be called antibody. You can call it antibody, that is AB, or AB, sorry. You can call it antibody AB, or you call it immunoglobulin, or you call it gamma globulin. So this gamma globulin now, we have already stated where we saw the plasma protein at the gamma, hence the name gamma globulin. For this immunoglobulin, we say there are those globulin which have immune function. Immunoglobulin, they are those globulin which have immune function. That is why we say antibodies are immunoglobulin or gamma globulin. They are immunoglobulin because they are those globulin which have immune functions. They are gamma globulin because they are located at the gamma region. They are located at the gamma region of the globulin in the plasma protein. So now, knowing all this concept, or with all these things I have stated here, let's define antibody in a beautiful way. I have already stated all these concepts. So if I may now ask you, what is antibody? So you can say antibody are those protein molecules which are secreted by those differentiating B cells called plasma cells. Antibody molecules specifically react with antigen, which stimulates the formation of those antibody. So what did I just say? Antibody are those protein molecules that are secreted by those differentiating B cells called plasma cells that react specifically with an antigen which stimulate the formation of those antibodies. You can as well define it in a way that antibodies are Y-shaped gamma globulin plasma protein produced by differentiating B lymphocytes called plasma cells. Each antibody is made to fight a specific antigen. So you should know that each antibody fights a specific antigen. There are trillions of antibodies in an individual, providing the body with the means to handle a specific antigen. So there are lots of trillions of antibodies in each individual. Now, we have gotten the definition of this antibody. Let's move into the structure. Let's get to learn about the structure. Remember, we stated that the antibody is Y-shaped. So I said it's Y-shaped gamma globulin. So the structure, what do you think about the structure? The structure gonna be like Y-shape. So the structure of antibody should be Y in its shape. I brought in a beautiful picture of antibody here. I will like to illustrate this diagram with my free sketching. Remember, I said antibodies consist of 4 interconnected polypeptide chain arranged in a Y shape. So these 4 interconnected polypeptide, they are arranged in a Y structure. So antibody will be having a shape like this, a shape that is like Y structure. The structure of antibody should be in Y structure. So this is the first thing you should know about antibody. Antibody has Y structure. Now this Y structure is made up of two heavy chain and two light chain. So this one I just drew here, this is the heavy chain, two heavy chains, this is the heavy chain, and the light one, I will show you where the light one is. The light one, they are just here, the kappa and the lambda, they are just here. This is the Y shape. So antibody has two heavy chains and two light chains. The light chains are just on top of this region, so it is not okay. The two light chain are towards this, look at it, they are just here, while the heavy chain runs up and runs from up to down, Now my question is, which area of this antibody engulfs the antigen? Which area of this antibody, which part of this structure holds the antigen? When the body notices an antigen and releases its antibody, what part of this antibody holds the antigen, the foreign body that enters the body in the body? I know you may be thinking is towards this large part, this wide area. No, the antigen is not there. I will show you. The area where you find the antigen, when the antigen comes into the body, the area that holds the antigen is this area. So the antigen can have different shape, and it is being held towards this region. So if I ask you what region of this antibody holds it, then you say is the epitope. The epitope is this area touching this antigen. The epitope is the area that touches the antigen. The epitope holds this antigen in this area. Then the next thing you need to know about the structure of this antibody is that it has a hinge region. Look at this diagram I imported. Look at this diagram here, this beautiful diagram. Can you see the cursor? This diagram at this region, look at the hinge region written here. So we have the hinge region. The hinge region is located at this area. It holds these two heavy chains together. It holds these chains together. It holds all this upper region with this down portion. It holds it together so that when this antibody changes its shape or molecule and spread wide apart, it will not get to be deformed. So this hinge point holds it from falling off. In this structure of this antibody, we can see that these upper parts of this antibody, we call it the fragment antibody, FAB region. Then this lower part, this down part here that has only the heavy chain, we call it FC. Fragment crystallize. So what did I just say now? I said this antibody structure, it has two heavy chains and two light chains. This one I highlighted with a red marker. The heavy chains are longer polypeptide chain. They are, can you see it is so long? While these light chains, they are shorter polypeptide chains. They are very short. Look at it highlighted on red. And I stated earlier that we have this upper region that consists of the heavy chain and the light chain. We call it FAB region. FAB region. And the down parts, the down area, we call it FC region. Another thing you need to know that is that there are two light chain. These two light chain, we call it kappa and lambda. Kappa, you will call it this one highlighted on red. You can call it kappa, or you say is lambda. So each antibody has either two kappa or two lambda chain, but not both. So you can see antibody, like if you, let's look at immunoglobulin G. For immunoglobulin G molecule, you can say immunoglobulin has two kappa or it has two lambda light chain. You won't see antibody that has kappa on one side and lambda on one side. So it has either two kappa or two lambda on the upper region. Let's go looking at the structure. What bond holds this heavy chain and light chain together? What bond holds it together? The bond holding the heavy chain and the light chain together, we call it disulfide bond. Disulfide bond link the heavy chain and the light chain, as well as the two heavy chains together. So it links all this. You will see, okay, look at this structure here. Look at this disulfide bond. We know disulfide bond is towards this region. Look at the disulfide bond. Remember I said earlier that antibody is polypeptide consisting of proteins and carbohydrates linked together. They are glycoprotein. So I said, remember that I said that antibodies are glycoprotein. So this glycoprotein, they consist of protein and carbohydrate chain linked together. So look at this beautiful diagram. Look at the carbohydrates. Look at these carbohydrates in this area. So we can say the carbohydrates are located at this region. You can see the region where you can see your carbohydrates linked together. The carbohydrates linked together to the protein. Now, with all things being explained in the structure of antibody, let's review everything I've said again. I said at the variable region, this is the variable light chain. This is the variable light chain. The VL is the variable light chain, and this is the variable heavy chain at the opposite side. So this is the antigen binding site, or you call it the paratope. It binds to the structure of this antibody at the location called epitope. So the paratope binds at this location called the epitope. Now still have this at this opposite side. We have the variable chain and the variable light chain and the variable heavy chain. They are the light chains. Okay, now let's look at this diagram. It has the hinge region. Look at the hinge region. This is the hinge region. Let me use a black marker. So this is the hinge region. I said this hinge region is located between the FAB. This is located between the FAB and the FC, and it allows for flexibility. I said it allows for flexibility. So the function of this hinge region, this is the hinge region at this location. This location allows for flexibility of antibody. Now we have the disulfide bond. The disulfide bond is the bond that holds this light chain and the heavy chain. So you will see a disulfide bond holding these two structures together at this side and also at this very side, and it will also hold this. The disulfide bond holds these together. So this is the disulfide bond holding the structures all together. It allows for stability, to maintain the stability of this Y shape. Now let's take a look at, can you see this constant region? Take a look at the constant region. So at this constant region, look at the CH1 over here, the CH1 here, the CH2 here, and the CH3 here. These make up the rest of the antibody and they are less variable. In this heavy chain, at this location of this heavy chain, at this stem region, this H2 and the H3, they are the C region that determine the class or isotype of the antibody. What do I mean by the classes of antibody? That's the immunoglobulin. We have the classes of the antibody, that is, you can summarize it as game. Game. So G is immunoglobulin G, A is immunoglobulin A, M is immunoglobulin M, E immunoglobulin E, D immunoglobulin D. So is this constant region at this stem that determine the classes of antibody. Is this classes at the stem that determines the classes of the antibody. Okay, I hope I'm making it as simple as I can. I'm trying my best to make it as simple as I can. So look at this region, the FAB. This is the FAB we talked about. Okay, look at the FAB. This FAB region, they include the heavy chain and the light chain. Look at it on top, or this portion of the heavy chain. Okay, let me use the red marker. This FAB, this is the FAB. It is the variable region. All this region is FAB. It includes the heavy chain and the light chain. This is the part, this FAB is the parts that bind to the specific antigen. So it's the region that consists of the region that binds to the antigen. Now the FC, what do we mean by FC? Okay, there was scientists, they carried out an experiment using this is a flask. Sorry, I'll tell you how this FAB and FC came about. Okay, let's do this way. I'll tell you, let me shift this and illustrate it very well. Groups of scientists, they carried out an experiment. This is a flask, and in this flask, an enzyme called papain was introduced into this. Papain was introduced into this flask. This flask has different shapes of antibody, y-shaped antibody. So papain was introduced. This is papain. Papain was introduced into this flask. When after the introduction of the papain, an antibody was then introduced again into this flask. This is an antibody. This is an antibody. This is papain. So this is papain. Papain was introduced into the flask, as well as antigen was introduced into the flask again. Then when the antigen got into the flask, it's binded at the antigen binding site. Like we talked about, we said at the antigen binding site, the antigen was seen binding to the structures. So the antigen was binding to this antibody in this area. So the antigen was not able to bind to the stem region, and the stem region was seen falling off from this area. So the stem region cuts away. This stem region, it's cut away. The stem region was found at the bottom of this flask. So what do we now say? We now say that the fragment crystallized. So what do we now say? We say the fragment crystallized. So the stem region fell off from the antibody and it was found at the bottom of the flask. So we call that fragment crystallized. Hence the name FC. We call it fragment crystallized, hence the name FC. So the FAB are the antigen that binded to this antibody. So the FAB region are the location where the antigen binded to the antibody, hence the name FAB and FC. It was during a test carried out with papain and antigen introduced into the flask, which the antigen binded to the antibody at the antigen binding site, and it couldn't bind at the stem region. The papain cut off the stem region. The stem region was at the bottom of the flask. So we say that the fragments crystallize. Then the FAB is the one that binded to the antibody. So I think we're able to touch all this region of the structure. I just made it so simple. So thank you so much for joining me in this lecture. I hope you find it valuable. I look forward to seeing you all in the next one. In our next class, we're going to be talking about the classes of antibody, the structure, and everything about the classes of the antibody. So, see you in our next class. I hope you were able to learn this, and I'll be dropping some questions in this antibody to test your knowledge. You can turn on your notification bell to know when I upload the next lecture.